Hello students, I am Dinesh Udpi and we are studying gene technology, immunology and computational biology paper in zoology subject which is introduced for fourth semester under NEP scheme. And today's video we will discuss about screening and selection of recombinant colonies. And this is the last session for the first unit, that is first chapter, that is principles of gene manipulation. So what is screening? Screening and selection of recombinant colonies. After the introduction of rDNA into suitable host cell, it is essential to identify those cells which have received the rDNA molecule. This process is called screening. So we discussed in our previous sessions. So what is done in recombinant DNA technology? So the gene of our interest is inserted into a host cell, into a target cell using different techniques. So at the end, we want to identify the target cells, whether they successfully taken up the gene of our interest, whether they are having the recombinant DNA or not. So that process is called screening and selection. So what is the basis of this screening? Very simple. The recombinant cells will express the characters because we have introduced some new genes into the target cells. So those new genes will express some characters, and the cells which have not taken, the cells who fail to take up the recombinant DNA, they will not express the character. So based on this criteria, we are going to screen and select the cells. So there are many methods of screening and selection of recombinant colonies. So we will discuss some of the most commonly used and effective methods which are used in genetic engineering. So one very powerful method is insertion inactivation method, and it is also called blue-white colony selection method. So because based on the appearance of blue colonies and white colonies, we are going to screen and select. That's why it's called blue-white colony selection method. So it's a powerful method used for screening recombinant plasmid. In this method, a reporter gene LAC-Z is inserted in the vector. So this is the important point we need to remember. The vector which we are going to use to transfer the gene of our interest, in that one gene called LAC-Z gene, which is considered as a reporter gene, is inserted. You are already familiar, if you studied lac operon concept, then you know what is this LAC-Z. LAC-Z is a part of lactose metabolism operon, and this enzyme is going to produce, this gene will synthesize or will produce one enzyme called beta galactosidase, right? And what is important in this particular scenario is this gene, this LAC-Z gene, which produces beta galactosidase enzyme, it has many recognition sites for restriction enzymes. So that is the important thing. Even it is a gene which is having many restriction sites. So we can easily understand the significance. The presence of restriction sites will enable the introduction of foreign gene into this LAC-Z gene. So beta galactosidase breaks a synthetic substrate called XGAL. So in the laboratory conditions, this enzyme is capable of breaking one substrate called XGAL. So 5-bromo, 4-chloro, indolyl beta D-galactopyranoside. So that is a chemical. It's nothing but a sugar. So this substrate is going to be broken down into insoluble blue colored product. So if beta galactosidase is produced, and if X-gal is present in the culture medium, then this X-gal is going to be broken up by this enzyme into a product which is a blue colored product. So this is important. So presence of blue colored product means there is an enzyme. So we'll understand how it works. If a foreign gene is inserted into lactate, this gene will be inactivated. Therefore, no blue color will develop because beta galactosidase is not synthesized due to inactivation of lactate. So when we break the vector, it is going to break because we are going to use the endonucleases which will cut the restriction site present in the lactate, and we are going to insert the gene of our interest. So that gene will be inserted into lactate gene. Because the inserted gene is going to break the lactate gene, the product will not be formed. So what is that product? Beta galactosidase. So if there is no formation of blue colored product, that means there is no production of galactosidase enzyme. Why this galactosidase enzyme is not produced? Because the gene is inserted. So indirectly it is an indication that the gene is present, the gene of our interest. So the recombinant DNA is successfully incorporated. So this is the principle which we use in this technique. Therefore, the host cell containing rDNA, it forms white colored colonies on the medium containing X-gal, whereas the other cells containing non-recombinant DNA will develop blue color colonies. On the basis of colony color, the recombinants can be selected. Through this picture, let us understand it more clearly. So this is the vector, right? And this is the lactate gene. So here using endonuclease enzyme, we have cut this gene, right? And this is the gene of our interest which we want to insert here. So the same endonuclease enzyme is used here also. Now what happens if the DNA gets inserted into the vector, right? We call it as recombinant DNA. We already know about this. So now this is a recombinant DNA. You can see here, this is the vector and this is the gene which is inserted. Now what happened? Because of the insertion of this gene, the LAC-Z gene is broken. You can see here, this is a LAC-Z which is broken into two separate pieces. Now it cannot express the beta galactosidase enzyme. Now if the insertion of foreign DNA fails, now what happens? This is the vector which has not taken the foreign DNA. So you can see the presence of LAC-Z gene here. So this is a LAC-Z gene which is intact. Now imagine the cells which have taken up this one, the cells which have taken up the insert less vector, right? So here are the two bacteria. In one we have the recombinant DNA and in another we have the vector without the foreign DNA, so without recombinant DNA. What happens? Because okay, so these bacteria are transferred into a media. In this media, the X-gal will be there. So you can see here, this is a X-gal plate with antibiotic. I'll tell you about the antibiotic later. Now, because X-gal is present, this cell will not produce beta galactosidase enzyme. So therefore, the X-gal will be taken up by the cells. The X-gal will enter into the bacteria, but this X-gal will not be converted into a blue colored product because the gene is broken here because of the presence of foreign DNA. So therefore, this will develop into a white colored colony. Whereas this bacteria, because the gene is intact, it will take up the X-gal and the X-gal will be broken down into a blue colored product. So therefore this bacteria will produce the blue color colonies. So the colony, the bacteria which has the gene of our interest, it will produce white colored colonies, and the bacteria which is not having the gene of our interest is going to produce blue colored colonies. So now we know that the white colored colonies contain the recombinant DNA, and we can isolate them using different techniques. So this is the blue white technique, which is very powerful tool or powerful technique by which you can screen and isolate uh, the recombinant DNA containing bacteria. The same thing I have used here, but this is not necessary because we already discussed about it. But see this picture. Here you can see the white colored colonies and blue colored colonies in the X-gal medium. Now you know these white colonies, they are the ones which contain the gene of our interest. Another very basic and widely used technique is antibiotic resistant markers, use of antibiotic resistant markers. An antibiotic resistance marker is a gene that produces a protein that provides cells with resistance to antibiotic. Bacteria with transformed DNA can be identified by growing on a medium containing an antibiotic. Recombinants will grow on this medium as they contain genes encoding resistance to antibiotics such as ampicillin, amphenicol, tetracycline, or kanamycin, etc., while others may not be able to grow in this media. Hence, it is considered useful selectable marker. See, the technique uses a principle where the vectors which we select, they contain antibiotic resistance genes, right? So the vectors will be having antibiotic resistance genes, and using these vectors we are going to transfer the gene of our interest into the bacteria, right? So if vector is taken up by the cells, that means uh, the antibiotic resistance is present, right? So the bacterial cells in which the vector has entered, it will show the antibiotic resistance. So the bacteria without the vector, it will not show the resistance to antibiotic, right? So here using this picture I can make it more clear. This is the vector, right? And in this you can see this colored portion. Let us consider this as the uh, antibiotic resistant gene, right? Now we use this vector to introduce the gene of our interest. Now there are two possibilities, three possibilities actually, when we transfer them to the bacteria. Either the bacteria has not taken up any vector, whether recombinant vector or non-recombinant vector. So here there are two possibilities. You can see here when we use endonuclease enzyme and ligase enzyme, there is a cleavage and then there is a ligation. So during this process, the vector may have taken up the gene of our interest. So this blue colored structure you can see here, it has taken up the gene. So now it is a recombinant DNA. Whereas this vector, it has failed to take up the gene of our interest. So this is a recombinant vector and this is a non-recombinant vector. Now there are three possibilities. One possibility, when we try to transfer this vector into a bacteria during the course of gene transfer technique, either the bacteria has not, it failed to take up the vector itself. So there is no vector, whether the transformed or non-transformed, I mean whether the recombinant or non-recombinant, the bacteria has not taken any vector, right? And in another case, the vector has entered, but it may not be having the gene of our interest. So you can say this. So the gene has not entered. And now we have this third case where the gene of our interest is successfully inserted into the bacteria, right? So uh, in this method, you can easily understand when we grow these bacteria in a culture medium, right? So the bacteria without vector. Now this is a culture medium where antibiotic is present, right? So antibiotic will kill bacteria. We know that antibiotic will kill bacteria, right? So when we culture them in this media containing antibiotic, if no plasmid is present, because antibiotic resistant genes are not found in bacterial chromosome, they are found only in the vectors, right? So because there is no vector here, these bacteria will die, right? So once they are introduced into the growth medium, they will die. Whereas the bacteria which have taken up the vector, whether the gene is present, the gene of our interest is present or not, whether it is a recombinant vector or non-recombinant vector, in both the cases, here also and here also, right? So here the gene is present and here there is no gene. In both the cases, the bacteria will show antibiotic resistance. So antibiotic will not affect the bacteria. So when we grow them in a culture media, if they die, that means vector is not present in the bacteria. Very simple. If the bacteria succeed in growing in a culture medium which contain the antibiotic, that indicates the presence of vector. But there is no guarantee that that vector is a recombinant vector. So for that we use blue-white technique, right? So the combination of antibiotic resistant gene and blue-white technique. So we use both the techniques here to first identify the bacteria with vectors and then confirm the presence of gene by using blue-white screening technique. So like this, blue-white screening and antibiotic resistant gene technique, they are used in a combination. Next we have molecular techniques. Many types of molecular techniques are there which are more sophisticated and comparatively more accurate. So isolation of genetic material and gel electrophoresis, right? So when the bacteria are grown in a culture medium, right? So those bacteria are subjected to the electrophoresis, and by PCR we can multiply the gene of our interest. Okay, so electrophoresis is a separating technique used to separate different biomolecules with positive and negative charges. Here, if you have studied gel electrophoresis technique, you know that the DNA is extracted from these and then they are cut into pieces by using endonuclease enzymes, by using different endonucleases, and then they are run in a agarose gel. Okay, and by studying the band patterns, so we can conclude whether the gene of our interest is present in the bacteria or not. So this is a modern technique which is more accurate. Okay, so here the same thing I can explain with the picture here, right? So this is a vector, okay, where the DNA of our interest is not there. This is not a recombinant. and here the gene is present. So this is a recombinant DNA. Positive amplification confirms the insert's presence. Now for this gel electrophoresis technique, we are going to use dyes, or we are going to observe it under UV, and we use primers for that. So first we have a primer, okay, which indicates the presence of vector. So it is similar to the antibiotic resistance gene technique. Antibiotic resistance technique confirms whether the vector is present or not. The same thing here. Only thing is we are using a molecular technique here, right? And the primer, if this line after the gel electrophoresis is run, and if we study it, if we get this line, the presence of this line will indicate the presence of the vector, right? And here there is one more primer which will identify the presence of the gene of our interest. Okay, so you can say we have two markers, one for the presence of vector and another one for the presence of the gene which we have introduced. Okay, so here in this case the insertless vector will not be showing the marker for the presence of vector. Okay, and then this is insert carrying vector, only vector is present here. Okay, whereas this one is the one which is carrying the gene of our interest also. Okay, so here this is the line which indicates the presence of gene, and this is the line which indicates the presence of vector. So only this is present, means vector is present. This line is absent, means no vector. This line is present, means only vector. And along with this line, if it is also present, the second marker, presence of gene. So in the third case, we can confirm that the vector is successfully incorporated and it contains the gene of our interest. So this is gel electrophoresis technique by which you can screen the presence of gene. Now the gel electrophoresis plate which we have produced, it can be subjected to a blotting technique. Maybe you are familiar with different type of blotting techniques for DNA and RNA and protein. So different blotting techniques are there, like southern blotting, northern blotting, and western blotting techniques. Blotting techniques are used to identify the presence of the gene of our interest. And lastly, there is one new technique which is called genome sequencing method. So Sanger sequencing or whole genome sequencing, I mean, there are DNA sequencing methods are there, and here we are going to use that genome sequencing method, right? We know the sequence of the gene which we have inserted, right? And we are going to collect the DNA from the bacterial cultures, because we are going to we are interested in knowing whether these bacteria, they have taken up the gene of our interest. So we have done the recombinant DNA technology, we have inserted the gene of our interested in them, and we want to know whether that gene is successfully inserted into them or not. So the gene, the DNA is going to be sequenced. So there is a DNA sequencing, and once we get the sequence, we are going to compare it because we already know the sequence of the gene of our interest, and if we get the same sequence from the bacteria, that means that gene is located. So this is the most modern technique, and this is the most accurate technique. It is 100% confirmation of the presence of that gene. So like this, we have so many different type of techniques for screening and selection. So that's about for this video. So you can use the references, you can use the website. I have given the link for website for different selection procedures. So thank you.